Hi, everyone. My name is Ellen. I'm a PhD student at UNC Chapel Hill, co-advised by Will Valdar and Marty Ferris. Today, I'm going to be talking about how we used CC027, which is a new model of peanut allergy, a new mouse model of peanut allergy, to study the genetics of peanut allergy susceptibility. So food allergies are a growing health burden. Approximately 1 in 10 adults and 1 in 12 children in the United States are affected by some form of food allergy. Peanut allergy in particular is very common and very severe. It's the second most common food allergy in children. It affects at least 1% of the US population, and peanut is the most likely food to cause anaphylaxis and death. We know that the development of food allergy is influenced by genetic predisposition as well as environmental factors. So mouse models would be a great tool, of course, to study the genetics of food allergy susceptibility, but previously existing mouse models for peanut allergy have been unideal. So in order to develop peanut allergy, they had to be sensitized, which involves injecting them with peanut protein and powerful immune response enhancing adjuvants such as cholera toxin every week for several weeks. And then after that sensitization process, in order to elicit an allergic response, they had to be injected with intraperitoneal injection, which is an abdominal injection. And even then, only certain strains are susceptible, meaning some strains after the sensitization process don't respond to IP challenge. So if we could identify an orally reactive model that would be more relevant to human physiology, and it would allow us to study the therapies that alter the immune system of the GI tract. So enter the CC. As many of you, of course, already know, the collaborative cross of the CC is a multi-parent panel of recombinant inbred mouse strains. They started with 8 genetically diverse founder strains and did a funnel breeding system, followed by several rounds of brother-sister mating, so that in the end, each CC strain is inbred with a genome that is a mosaic of those 8 founder strains. So there's a lot of genetic diversity in the CC, comparable to that present in some human populations. And since the strains are inbred, it's a replicable system. And the genomes of these strains are very well characterized. So a common experimental design is to screen the CC for strains that exhibit a particular phenotype of interest. And due to the genetic and phenotypic diversity present in the CC, we're much more likely to identify a new model over classical inbred strains. So that is what Kelly Orgel of the Close Lab at UNC did for peanut allergy, for orally reactive peanut allergy specifically. She screened 16 CC strains for strains that can be sensitized and react orally to peanut challenge. So the sensitization process, as I described before, involves injection of peanut protein and cholera toxin as an adjuvant every week for 4 weeks. And then they are challenged with peanut protein on the 5th week. And crucially, that challenge is done orally because we're looking for orally driven susceptibility mechanisms. So these are the results. We're looking at temperature curves for 60 minutes post-challenge here. And that is our phenotype of interest because anaphylaxis in mice is indicated by an acute temperature drop. So on the left here, we have our non-reactors. They don't experience a significant temperature drop, and all of the classical inbred strains that have been studied fall into this category as well. In the middle, we have mild reactors, and then on the right, we have our severe reactors, which include CC027. We can also see that CC12 had a severe reaction as well, but on repeated experiments, CC12 did not react. So CC27 is our only strain that consistently and robustly reacts to oral peanut challenge. So we have CC27 that reacts to oral challenge. We have C3H, which was a previously existing mouse model for peanut allergy that required challenge with abdominal injection. So Johannes Meekins, also in the Close Lab, did a backcross between these 2 strains to identify genetic loci that are associated with that orally driven anaphylaxis phenotype. So they sensitized the mice as described earlier, and then challenged them orally with peanut protein with oral gavage, and then measured temperature for 60 minutes post-challenge as a measure of anaphylaxis severity. And we had about 360 backcross mice. So these are the temperature trajectories post-challenge. We can see a pretty wide range of responses. I did hierarchical clustering analysis on these trajectories and saw that these trajectories grouped into 3 pretty distinct groups. We have the non-reactors in pink, and then mice that react and then recover in green, and then mice that react and continue to decline in blue. So these mice were genotyped with mini Muga, and then I did QTL mapping on the temperature phenotypes. So all of the genome scans that I'm about to show are with respect to some aspect of the temperature trajectory. And the dotted line is the 0.05 significance threshold. So this is for temperature at 15 minutes. We have this peak on chromosome 10 that I named QPA1. At 30 minutes, this peak on chromosome 17 appears that I named QPA3. And then when I QTL map on the cluster from the previous slide as a phenotype, we have this peak on chromosome 5 that appears that I named QPA2. So these are kind of my three major QTL associated with anaphylaxis severity in this back cross. So I'm going to focus on QPA1, which is associated with temperature at 15 minutes. As many of you probably know, with QTL mapping and GWAS, it can be really difficult to find map and identify a candidate gene from the tens or hundreds of genes that are present in a particular locus. But in this case, we were able to identify a pretty convincing candidate gene. So that's what I'm going to talk about for the rest of this talk. And this is just showing the relationship between the genotype at the peak of QPA1 and the temperature at 15 minutes. We can see that mice that are homozygous for the CC027 allele at this locus have a significantly lower temperature, so more severe anaphylaxis than the mice that are heterozygous at this locus. So if you recall that each CC strain's genome is a mosaic of the 8 founder strains, I mentioned earlier that the genomes are very well characterized. So we have these haplotype maps where each color represents one of the 8 founder strains. So if we zoom in on chromosome 10, and we can zoom in on the QTL scan there as well, we can see that CC027 harbors a Cas and B6 haplotypes across this locus, which is highlighted in gray here. And this is interesting because strains that harbor Cas and or B6 at this locus, as well as most notably B6 itself, are refractory to peanut allergy, meaning even after that sensitization process, they don't react to peanut challenge. So we thought that CC027 might have a private mutation that's inferring the susceptibility. So these particular strains have been inbreeding at UNC for over 10 years. A private mutation occurs when a de novo mutation is fixed within the population. So we thought that a private mutation on top of this Cas or B6 background might be inferring the susceptibility to oral peanut challenge that is specific to CC027. So a group at UNC and JAX did whole genome sequencing of all of the CC strains and identified several private mutations across the CC. So we referenced this list and found that CC027 does have a mutation right near the peak of QPA1, which is intronic to the gene Themis. So Themis is a gene that's involved in T cell development. T cell development is the process by which mature T cells are produced in the thymus. So starting with these lymphoid progenitors that come from the bone marrow and migrate to the thymus, they are turned into immature double negative T cells that are turned into double positive T cells, meaning they have both a CD4 and a CD8 receptor. And then those are turned into mature single positive T cells that are either CD4 positive or CD8 positive. So those are the mature T cells that are exported to the peripheral lymphoid sites like the spleen and the lymph nodes to perform their immune system function. So why is it interesting that this gene, we think this gene that might be involved in peanut allergy is involved in T cell development? Because CD4 positive T cells are the primary orchestrator of the allergic response. So some problem with how these cells are developed might be related to peanut allergy development. And Themis is highly conserved across mammals, so it's an interesting candidate gene to study. So we designed an experiment to test whether this private Themis mutation might be affecting CC027's T cell composition. So we identified 4 CC strains that have a B6 haplotype at this QPA1 locus, but of course without the private Themis mutation. And then we measured the concentration of several T cell subtypes in the thymus, the spleen, and the mesenteric lymph nodes, which are the lymph nodes in the gut that are most relevant to food allergy. And we measured the overall abundance and relative proportion of several T cell subtypes in those organs. And we found that CC027 does have a significantly different T cell composition from these other strains. So in all of these box plots, CC027 is on the right. So we can see that CC027 has significantly more double negative T cells in the spleen. Again, these are immature T cells that shouldn't be making it out of the thymus in very high amounts. So seeing significantly more of those in the spleen indicates aberrant T cell development. We have significantly fewer CD4 positive and CD8 positive T cells in the spleen. So those are mature T cells. Again, seeing fewer of those indicates aberrant T cell development. And then really interestingly, CC027 has significantly fewer regulatory T cells or Tregs in the spleen. So these are a sublineage of CD4 positive T cells. They're involved in tampering the immune response so that the immune system doesn't recognize things that shouldn't be harmful as harmful, like food and ourselves. So decreased Tregs has been implicated in allergies and autoimmunity. So it's really interesting to see this phenotype. So in summary, we used a CC027 by C3H back cross to identify loci associated with orally driven anaphylaxis severity. We identified Themis as a candidate gene for one of these loci, QPA1. Themis is involved in T cell development, and CC027, which has a mutant Themis, has a variant T cell composition compared to other strains with the same haplotype at this locus. Given the role of T cells in the allergic response, we think Themis is a plausible candidate gene driving the association at QPA1. Also, this study highlights the utility of the increased genetic diversity of the CC in improving animal models of human disease and studying the genetics of disease susceptibility. So I'd like to thank my labs, the Ferris lab and the Valdar lab, our collaborators, especially Mike and Johanna from the Berks lab. We just submitted this, so if you're interested in seeing the preprint, you can scan this QR code. Yeah, so I'll take any questions.